Good morning, everybody. I'm a little late on the upkeep of posting this video. My goal was to post it yesterday. Completely my fault. You guys are probably in convocation ceremony right now, and I just want to say congratulations for making it through the first exam. Right now, we're going to get into it and go through the nitty-gritty of Chapter 4. We're on the last part, so let's go ahead and get started, guys. So from the objectives, I think the only things we've already done so far is, if I use my little pointer, we've explained the cell theory, which remember there's two tenets that you need to know. Sorry, this blurry background's weird. Whether something is living or non-living. Remember, viruses are non-living, and they cannot reproduce on their own. The difference between structures of viruses, prokaryotes, eukaryotes. Again, prokaryotes are unicellular, have circular DNA. Eukaryotes are multicellular with linear DNA. Viruses are non-living. Prokaryotes and eukaryotes, like your bacteria, are your prokaryotes. Your plant and animal cells that are your eukaryotes, those are living. And then viruses, of course, are non-living. Diagram a plasma membrane. So we kind of went into the nitty-gritty of that as well. So remember, it's a phospholipid bilayer with polar hydrophilic heads and non-polar hydrophobic tails, okay? So you want to know also the selective permeability, which has to deal with passive and active transport. So remember, active transport, think of active transport, it's got to get active, it's moving around, so it needs ATP. And then passive transport doesn't need ATP, but you've got simple diffusion and facilitated diffusion. Remember that diagram I posted on Blackboard that we did together? So simple diffusion, just going down the concentration gradient. Osmosis is an example, doesn't need any aid. Then you've got facilitated diffusion where it's still down the concentration gradient, high to low concentration, but you need like receptors and stuff to help with that. And then active transport is going against the concentration gradient or up the concentration gradient. So that's going from low to high concentration. And that has to deal with like using ATP. So the sodium potassium pump is a perfect example that we saw in that video. And then now we're going to get into these last three. By the end of this lecture, you will have done all the enabling objectives, but these are the three we're doing today, okay? So bear with me, guys. You know I can't draw, so sorry. Okay, so we're going to finish up with the membrane, the plasma membrane. So remember I was talking about viruses, how they don't just diffuse across the membrane, they break in. So remember I kind of drew that structure. Oh, Lord, give me strength. This is the cell. Yep. And then this is your virus right here. And these are its little spikes, I guess, to latch on the membrane, and it breaks it. It invaginates and breaks it. It doesn't just spread evenly because it's a pathogen that's not even supposed to be in the cell. So it finds its own way to break in. And I kind of showed that yesterday, so we're going to get into that more so today. So COVID-19, SARS-CoV-2 enter human cells with the spike proteins that I talked about. Remember coronavirus we said was named after crown, based off the shape of its spike proteins that are on the receptors of its membrane, and it allows it to kind of break in. And it binds to the membrane protein ACE2, which is a membrane protein that is on your plasma membrane. And what it does is it's an enzyme on the membrane, and it cuts up larger proteins into smaller usable units. Because remember how we said there are certain things that can go through. So obviously small molecules or hydrophilic molecules are able to easily go through the plasma membrane. But large molecules or hydrophobic molecules cannot easily go in the membrane. So what ACE2 does, it's an enzyme that's attached to the membrane protein on the membrane, the plasma membrane. And when the coronavirus attaches to ACE2, it uses the ACE2 enzyme to break it down into smaller usable chunks. Because again, virus is a big, large molecule that can't easily get in. So it uses ACE2 to kind of break it down, make it smaller, so it can kind of break in and invade into the plasma membrane easier. And actually, the research we talked about on Tuesday, Gobel, yep, right here, she was the one who actually found ACE2 as that enzyme that kind of helps coronavirus break into our cells. So yes, the coronavirus spike binds to ACE2 and allows that fusion into the membrane. And then once it's in the membrane, remember we talked about that? This virus breaks in, so it's in the cell, and it's making these little, we'll make the green its DNA. It fills the cell with its DNA and uses the host cell's organelles to replicate it, and then cell bursts, boom, you have more viruses to spread, and that's how that happens. And then remember we said that it was this mutation, D614G, so aspartate at position 614 changes to glycine. That's that mutation for the Omicron virus that she had researched. I'm not going to ask you to know this mutation because that's honestly biochemistry, but I will ask you, I won't even ask you the specific enzyme ACE2, that's again some more biochemistry, but I will ask you the mechanism of how a virus breaks into a cell, invades it, breaks down, and then replicates. And we're going to get more into that today. So vesicles are kind of packages. So I think remember how we said the cell has several compartments to it, right? And these compartments, these organelles have different roles for the job. Vesicles are like your packages, and we use these vesicles to kind of go in or leave the cell, for particles to enter the cell or leave the cell. So entering, when you think enter within, that's endocytosis. So they use vesicles to bring stuff, molecules, whether there is any type of particles, get into a vesicle, and then they use endocytosis to go into the cell. Now, exocytosis is the exact opposite. Exocytosis expels or causes stuff to leave the cell. So if you have certain particles, and a good example of this is your lysosomes, which we're going to get into all the organelles in a minute, but lysosomes will break down certain material. And I think of lysosomes as your trash bags. They're the trash compactor, and then the vesicles are your trash bags. They kind of take all this crap from this waste material the cell doesn't need, puts it in a vesicle, which is what we're talking about right now, and that vesicle gets to the plasma membrane, fuses with the membrane, and then pushes the material out. So pushing the material out from the membrane is exocytosis. Bringing the material in from the membrane, that's endocytosis. So it's really important you know those two different. So now to go more into endocytosis, obviously I kind of belabor the point, you know me. Cells will take substances in through endocytosis and move them out through exocytosis, yes. The biggest point I want to draw back on this, I could ask this on the test, what are some forms of active transport? And, you know, the sodium potassium pump, I gave that as a good one. And remember, so active transport requires energy, ATP. Both endocytosis and exocytosis are forms of active transport, okay? So that's a very big thing to know. And it makes sense because you need a vesicle, you need energy to push it in or push it out of the cell. So endocytosis and exocytosis are those forms of active transport, not passive, okay? And of course, there are 3 types of endocytosis, and I need you to know all of them. Sorry, guys. So we've got receptor-mediated endocytosis, phagocytosis, and pinocytosis. And I'm going to give you little pearls for each one. So for the biggest thing, receptor-mediated is selective, and it's a selective process, and it only allows certain molecules to get in. So when I think of receptor-mediated endocytosis, those receptor proteins that are on your cell membrane, let's pretend the cell membrane's a door. And sorry for that loud noise outside my apartment. But the cell membrane's a door, right? And you want to try to get in. And the door's got, you know, like key cards that allow us to get into the lecture halls, passes allow you to get into certain rooms. It's got a little sensor. That's the receptor-mediated endocytosis, right? That's that receptor-mediated protein right there. And receptor-mediated endocytosis is when there's the proper key card that will let you into that door. So it's very selective. It's a selective type of endocytosis, a selective type of permeability, okay? It's very important you know it's selective. But basically, receptor proteins recognize that certain signal. And they're like, for example, aquaporins allow water to get in. And so they see it's a type of receptor protein that allows water to enter the cell. So an aquaporin, if it sees a water molecule, it kind of receptors, it goes, oh, wait a minute, you're water, you can come in. And there's other instances of receptor-mediated proteins, but for example, if there's like a protein and there's like a, you know, I guess if it's a muscle cell, for example, and you need more ATP to get in or more energy and stuff, it could signal that and see that at the membrane, and the receptor-mediated endocytosis will create a vesicle, push it in, right? But it uses receptor-mediated proteins. So that's the big one for that. And remember, it's selective. Now, we have phagocytosis and pinocytosis. So the best way to remember this, for medical terms, when we talk about phagia, That's like eating. So phagocytosis is cellular eating. So basically, it's a type of endocytosis of a large substance, and it brings things in and engulfs them. So I think cell eating. A big pearl that I want to give you guys on phagocytosis, which I'm going to do right now. Yes, I'm going to look up my textbook. You know, my textbook has little test questions I've already made. When it comes to phagocytosis, it is it selective? Yes, it is a selective one. I'm trying to look, without giving you the test question, I'm looking at the test question right now. What are 3 types of endocytosis? I'm probably going to put that on the test. And then I might ask, what's an example of receptor-mediated endocytosis? Here it is right here, LDL. So this diagram, if you go into your textbook, I got this from the textbook on page 74, but low-density lipoproteins bind to the receptors and transport them into the cell. So that's an example of receptor-mediated. Phagocytosis, an example of that for cell eating, you can think of yeast coming into the environment, and again, phagocytosis is selective, which also makes sense. So when you think about it, selective, you got to work a little harder for that, right? And phagocytosis allows large molecules to get in. I don't want to work, that's a lot of energy to put that, or for example, a lot of work. I don't want to do that for every single large molecule. So phagocytosis is another selective type of endocytosis. So that's important too. It's selective and it's cellular eating for large molecules. Those are 3 big things you want to know for phagocytosis. Now, penocytosis is different. It's non-selective, so that's important. So I could ask you, of the 3 types of endocytosis, which are selective and which are non-selective. The selective ones are receptor-mediated and phagocytosis. The non-selective, which I'm going to do in green, is penocytosis. It's non-selective. It's a lot less work to do because it's bringing in liquid, and it's called cellular drinking. So think of Pinot Noir. You drink, I'm a wine girly, sorry, but you drink Pinot Noir, cell drinking, right? And a good example of this is, outside environment. You see this right now, the fluid-filled vesicles. That's a good example. Again, check out page 74 is a very good visual for this, and that's kind of what I used here. So that's pretty much anything I could ask on this slide. Now for exocytosis, remember I said endocytosis is a vesicle, attached to the membrane, got the particles, and it brings the stuff in. Exocytosis does the reverse. You've got these vesicles go to the cell membrane. They're in the cell right now, and these particles leave the cell for any number of reasons. It could be just its waste product that we don't need anymore for our cells, or it could be for cell signaling. We need to bring stuff from one cell to another. That happens a lot with nervous cells, which if you take anatomy physiology, we use exocytosis to go from one neural synapse, chemical synapse to another. That's how you do brain signaling for like a thought to happen. So there's multiple reasons for exocytosis. But what you need to know for this class, exocytosis is contents are discharged to the exterior. So, and again, it's also a form of active transport, but it is leaving the cell. And it still fuses with the plasma membrane, but it's discharged out of the cell. Okay? And now this is just another visual of everything I went over. Again, definitely take a look at page 74. I love this example right here. That's all I'm going to say. You know what I mean by that. But I love this example right here. Page 74, guys. And sorry, I know my handwriting is horrible. I'm trying to use it on computer, but. All right, so yeah, we kind of, again, Dr. Gobale, she was studying coronavirus, trying to figure out the different mutations and strains. And she used cryo-electron microscopy, which is cryo-EM, to kind of look at different mutated versions of the spike proteins. And again, remember I said spike proteins, if I'm drawing coronavirus, here's the little virus, and here's its little, it looks like this. It's spike proteins that are on its membrane outer exterior that allow for efficient attachment to our host cells. Oh my goodness. I really am not an artist. But yeah, these are your spike proteins. So she noticed that there was a mutation, because remember we had, if you guys remember coronavirus, which I remember COVID-19, that was like a fever dream, but you had the first strain, and then we got vaccines out, and then around 2021-ish, 2022, there was another outbreak called Omicron, And even with the vaccine, people were still getting sick, and we're having these various like recent deaths too. So they need to do another update of the vaccine, a booster shot. That's why you get a booster every year, not just with COVID, but flu as well, because of these mutations. And if you take evolutionary medicine later, we can go into that. I'm not going to cover that. But the importance on this is for mutation rate. Dr. Gobale noticed there was a mutation that caused 2 small changes in the spike shape, which made it easier to bind and enter the cells. Now, this happened after the COVID vaccine, the initial one, right? The initial Pfizer one that came out. And there is, although a lot of the way it works with viruses and stuff, I'll just give you like a layman's term on it. You know, we have a virus, and then researchers come up with an antibody or with a type of vaccine to fight that virus, and it usually kills off, I want to say like 97% of it, right? But then there's that 3% of viruses that survive, and not only do they survive, they mutate and thrive to fight against that initial vaccine, so they can no longer be killed off. And those are the ones that found, Dr. Gobale noticed they had a slight change to their spike shape from the original variant. And so those are the Omicron. And again, variants are different viral strains. I know I'm saying that, and you guys are like, what's a variant? For COVID, for example, we have the Alpha variant, the Omicron variant. Those are different strains that mutated from its original one. And we also had Delta. It was Alpha, Delta, and Omicron, I believe, yeah. And Delta variant improved the permeability into the cell with that spike variant, like I talked about. So with the spike mutation from Alpha to Delta, Dr. Gobale noticed there was a mutation change, so a physical change in the spike proteins, making them easier to break into the host cells for Delta. And then to Omicron, it was even more vicious and was able to kind of break into the cells. Okay, so how a virus hijacks the cells. Usually, and I said this from Tuesday, viruses could be airborne, water droplet. COVID was an example, could be spread airborne through water droplets. Like if I sneeze, if I have COVID and I'm not wearing a mask, which is why we said to wear a mask, and I just cough and sneeze in the air, I have just spread millions of COVID-19 particles and water droplets that can enter the air, travel very quickly, and get into somebody's system, right? And through the ears, nose, throat, sinuses. And so it passes easily, yeah, from human to human in the air. That's called airborne disease. And then another reason why COVID was so deadly is you have some people, if they didn't have pre-existing conditions, and I was an example of that this is back when I was in the Navy when COVID came out, and I was on underway in deployment, and I was one of those that was diagnosed with COVID, but I had no symptoms. But again, just because I didn't have symptoms, I was highly contagious. And so you could be symptomatic, like asymptomatic, which means you don't have symptoms for days, and just be spreading it by just talking, breathing and stuff if you don't have your mask on, and you're getting other people sick without realizing it. And you could be contagious before symptoms appear was the point of that, which is why we kind of pushed on wearing masks, because even if you have it and you're like, well, I'm not sick, great, but you could be giving it to people that have pre-existing conditions, making them sicker. And yes, can be deadly, especially for elderly and those with compromised immune systems. All right, so in order to understand how a virus like COVID-19 can enter a cell, hijack its cellular structures and organelles, and use it to create more of its own virus before using it up and killing the cell, we need to go into each organelle. So the first organelle I want to go into is fluid-like jelly substance known as your cytoskeleton. So the cytoskeleton is kind of a network of protein cylinders and filaments, and they form the framework of your cell. So a lot of eukaryotic cells, they'll have cytoskeleton. I think of skeleton. They form a skeletal structure. They have 3 main types of fibers known as microfilaments, microtubules, and intermediate filaments. I'm not going to go deep in the weeds on what each one is, but they are basically aiding into the cell structure of the cytoskeleton. Okay, microfilaments have actin, and then intermediate filaments. What you need to know is they have different structures that aid for the cytoskeletal structure. And also, both plant and animal cells do have cytoskeletons. Plants also have a cell wall on the outside to even give more rigidity, but both cells need cytoskeleton. And in the cytoskeleton, that fluid material in between is called the cytosol, and it's just fluid jelly-like substance. And yeah, that's pretty much it. And they allow for cell movement and also give shapes to cells even if you don't have cell walls. So for animal cells, we don't need cell walls because we have the cytoskeleton, we have these filaments, microtubules, and we have that jelly-like structure to kind of hold stuff in place for the organelles. Which is why these organelles aren't just floating around aimlessly. They're in their set positions for a reason. 3 main types of cytoskeletal fibers. Okay, so I already went into them. You guys know it's those 3. I could ask you that. What are the 3 types of cytoskeletal fibers? Your microfilaments, your microtubules, and your intermediate filaments. The biggest point about these is interactions among these 3 fibers will help maintain cell structure. And they also permit changes in the cell shape to allow for like cell motility, like fluidity. And what happens with viruses is they will actually rearrange your cell microtubules and help them move around the cell so they can push them aside. So think of like if I'm a virus, and let's say I'm swimming in the water, for example, and there's like a bunch of pool noodles and stuff in the way, and let's say those pool noodles are your microfilaments and stuff in your fibers of your cytoskeleton. I, as a virus, can push those out of the way so I can get to where I want, which is mainly the ribosomes and stuff in the nuclear envelope, so I can double up my genetic material. So I will actually be like bullying, pushing cytoskeletal structures out of the side, which also inhibits the host cell, because as I'm doing this, I'm moving it around, changing it, and so that's pretty detrimental. So the cell wall. Cell wall is, again, like I said, it's for plant cells only, and it's outside of the cellular membrane, and it allows for rigidity and more structure, which we need for plants. And it was, only for bacteria, fungi, and plants. It's not going to be in animal cells. Animal cells only need a cytoskeleton. So for the endoplasmic reticulum, so there is a smooth ER and a rough ER. So if you can see it right here, this is the rough ER because it has ribosomes. Smooth ER does not have ribosomes. They each have 2 different roles. Smooth ER is for lipid and hormone synthesis or production, and rough ER is for protein production or synthesis. And also smooth ER can be for transport of some ribosomes if needed. But for the most part, if I ask you something, the smooth ER is for lipid and hormone synthesis, rough ER, protein synthesis. And it's mainly just a network of sacs for the outer membrane, and it is contiguous to its nuclear envelope, which is contiguous to the nucleus. And it is just like that. So what I mean by contiguous, I mean next to and touching. You've got this rough ER, smooth ER, and then you've got the nuclear envelope, and then you've got the nucleus. They're all next to each other in that order on purpose. Because when it comes to transcription and translation, which is how we make genetic code and proteins, it starts in the nucleus, then goes out to your nuclear envelope, to your ER. So it actually goes this way, which is why these structures need to be next to each other, by the way. Again, structure means function. So then, for how a virus attacks a cell, again, ribosomes, cytoplasm, they carry out protein synthesis for the rough ER. Proteins and lipids made by the ER are packaged into transport vesicles so they can be used. And yes, the biggest thing I want to say is the endoplasmic reticulum is contiguous to the nuclear membrane. It's next to it. And it's kind of almost impossible when you're looking at a cell to see where the nuclear envelope ends and your endoplasmic reticulum begins, because they're right touching each other on purpose to allow for that DNA replication and translation into proteins. So when it comes to COVID-19, it would take its RNA genetic material. Remember I said I'm going to tell you how it replicates it? Once that virus of COVID-19 uses its spike proteins to break into the cell membrane and come in, and then it starts floating away, pushing cytoskeleton out of the way, because it's trying to get to the ER, because that's where we make proteins, and that's how it's going to make more viruses. So it expels its genetic material and then uses the endoplasmic reticulum and the ribosomes to create more viral proteins. It can make up to 29 different types of proteins in that cell, and then expels its genetic material, makes more, multiplies, and that is how a virus uses our cell against us. So when it comes to transport vesicles, remember I said you have vesicles that are, kind of think of them as your packaging material. Think of your vesicles as the boxes that you're going to mail something, right? Like I got an Amazon package, I ordered it, and I needed to get delivered to me. The Amazon package itself is the vesicles. The Golgi apparatus is the Amazon transport truck sending things off. It is your mailbox. Golgi apparatus is the one that's sending stuff off, the delivery service. I like to think of it like that. So that's what your Golgi bodies, your Golgi apparatus does. It repackages proteins and lipids into those vesicles so they can be exocytized out of the cell or just into different compartments of the cell. Now we've got our lysosomes. And it's always kind of nearby your Golgi apparatus on purpose, because anything that the Golgi apparatus doesn't need, it's going to send to the lysosomes. And I think of lysosomes as your trash compactors, and those will break down the material with enzymes and then expel it. It's like recycling centers of the cell. And it will release those breakdown products into the cell interior to be discarded or reused if we need it. Lysosomes are not in your plants or fungi. Those are called vacuoles instead. Does the same thing. You've got little vacuoles that do the same thing as a lysosome, and then you have your central vacuole that does the same role as a lysosome to break down material, but also retains water, like I said on Tuesday, for fluid and rigidity for the cell. So the big thing you need to know about lysosomes, they break down material. Something close to lysosomes, which I'll get into in future slides, is peroxisomes, and it has hydrogen peroxide, and it uses it to break down fatty acid material through beta oxidation, and that is used so that you can make the phospholipid bilayer of your cell membrane. That's the importance of peroxisomes. They're also involved in the pentose phosphate pathway. You don't need to know that. The biggest part of peroxisomes is, just like lysosomes, they break down material with hydrogen peroxide, but they break down fatty acid material so we can make more phospholipid bilayer. So you've got your lysosomes, which is your trash compactors, your peroxisomes, also trash compactors, but once they break down those lipids and fatty acids, they make a cell membrane out of them, okay? And then, this slide is just going again onto the virus, the external components of everything. And most of these viruses will form an envelope by taking those kind of materials and particles of the cell's edge, budding out, and take some of that cell's plasma membrane with it, which we talked about on Tuesday. Remember, it has its own like lipid membrane, and it gets it from the cell, the host cell. Vacuoles, I'm not going to belabor the point, but again, vacuoles only in plants, fungi, some protists. They're for water balance and storage, and they could also break down noxious compounds and stuff. They also can contain noxious compounds that can kind of deter herbivores from eating them, so it's a protective mechanism too. When I say noxious is like, odorous or maladaptive or toxic compounds for your animals. So now we're going to get into one of the most well-known organelles, the mitochondria, which is the powerhouse of the cell. Yeah, remember I said that? Okay. Basically, the importance of it is that they transform energy from our food molecules or anything, or solar energy for plant cells, and we make ATP through a process called cellular respiration, which is found in all eukaryotic cells. That's the biggest thing. Mitochondria and chloroplasts. So plant cells have chloroplasts, and they also have mitochondria, but they also mainly have chloroplasts that will take solar energy and make it into ATP as well, because plants use photosynthesis. Animal cells only do cellular respiration. But still, the point of mitochondria is we take energy and we take kind of food molecules and stuff like that, and we make energy out of it that we need to use. The biggest thing about the mitochondria is they have an inner membrane, which have cristae, and an outer membrane. The purpose of the outer membrane of the mitochondria is it kind of makes a barrier between the inner membrane and like the cytoskeleton and everything like that. The inner membrane that has cristae also have ribosomes, and its infoldings. You see how it's kind of folded inwards like that? It looks like these little dashes right here. That's so that we can increase surface area for the electron transport chain. So it allows more area to make more energy through cellular respiration. That's the biggest part you need to know about that. And then chloroplasts, like I said on the last slide, your chloroplasts found in cells of plants and some protists, and kind of do the same function as mitochondria, but for photosynthesis. So they take that solar energy to make it. These are some symptoms of long COVID. So after a while, Dr. Gobel noticed that there were some symptoms that were remaining throughout, even after you were done with COVID in your body. It would be chronic exhaustion, pain, brain fog, and these persisting symptoms that we have, researchers noticed that this was due to impaired mitochondria, which makes sense because mitochondria creates ATP and energy that we need to move, do daily tasks, even thinking. We need like mitochondria for our nerve cells. So when those are impaired due to COVID damages our mitochondria, then we can't move as efficiently, chronic exhaustion. We can't recover as fast, so chronic pain. And then obviously I'm getting like spacey and stuff, brain fog. So that's, again, lower than normal ATP levels due to damaged mitochondria from COVID. The nucleus is your brain center. The nucleus is the brain of the cell. It's got, it's the control center of the eukaryotic cell, is a good way of saying it. And it contains your chromosomes, your histone proteins. It contains the DNA to make proteins. And it is enclosed by a double membrane called the nuclear envelope, which we said is contiguous to your endoplasmic reticulum. Remember that? And it's got nuclear pores, these speckled nuclear pores that allow chemical signaling to go in and out of your cell. So when it comes to viral infections like COVID, what will happen is, if you wonder how sometimes some infections in your body are worse than others, a lot of time that also has to do with how they're affecting your nucleus, because some infections will get to your nuclear pores, like this nuclear envelope, and they see these pores that are speckled, and they will clog them. And when they clog them, that doesn't allow for the chemical signals to come out of your nucleus and therefore out of the cell. Now, why does that matter? Your cells will send out a message to other cells whenever it's being attacked by an infection to tell your antibody cells, hey, come help me and attack this pathogen so we can get better. When a viral infection or some type of bacterial, any type of pathogen invades your cells and it latches onto the pores and clogs them, what happens then is that pathogen is not letting your nucleus send out those chemical signals of SOS, help, please, we need help, come on, bring in the antibody, kill this pathogen. And if that's not happening, then that pathogen's getting stronger and will not die as effectively. And that's how that works. Yeah, we'll block those nuclear pores. The double membrane bound nucleus is the hallmark organelle of all eukaryotic cells. It is double membrane bound, by the way. So yes, we went over Omicron, takes over the cells, replicates with a unique speed and magnitude, because again, it's mutated, so it's more efficient. Multiple vaccines were produced within a year of the emergence, and it usually takes much longer. It was fast because of the overlapping of vaccine development. You had multiple companies doing it, and just billions of dollars was kind of flooded in for this. But the Omicron variant emerged with more spike protein mutations, and it was more transmissible and able to evade protection by the vaccines. Because again, I said it kept mutating, so those spike proteins mutated to attach to the cell membrane easier. Some of this COVID-19, the Omicron variant would rush to the cell, to the nuclear pores, like I told you, attach to it and be like, nope, you're not going to send a signal out to get help. I'm killing this cell, and I'm using it to make more virus. So there was a drug that blocked the enzyme of the virus that needed to replicate it, and that was part of the vaccine to kind of help with, dissipating COVID. So we have gone in depth on all of our lecture. I know that was a long thing. We have finally finished chapter 4. So what's going to happen now? On Tuesday, we're going to do a quiz before we even start chapter 6. I told you the quiz is going to be a Kahoot game. So try to study up this weekend. Do your smart work. I didn't say that, but you have chapter 4 smart work. You have chapter 4 smart work, and then you also have chapter 6 inquisitive. Don't worry on chapter 6 inquisitive yet. Again, the homework that you have for smart work and inquisitive isn't due until test day, which is October 3rd. So you have 2 weeks to do it. But what I recommend is not just to get ahead, but also help with studying and efficient studying if you want to do well on the test. I would go ahead and read the textbook. Yes, read chapter 4. It's like 20 pages, guys. And then I would go ahead and do your smart work this weekend. That way, any questions you have, you can come to class on Tuesday and we can ask them. And also, we're going to take the quiz. And remember, the Kahoot game, as long as if you come to class, you get 100 on the quiz because you're going to participate in the Kahoot. But the way it's going to work is number 1 gets 10 bonus points on their exam. Number 2 gets 7 bonus points. And number 3 gets 5. And then we're going to take the quiz, and then we're going to go into chapter 6. Then we're going to finish chapter 6 next Thursday, and then we're going to take another quiz at the end with the same thing. First place gets 10 points. Second place gets 7 points. and third place gets 5 points. So if you study efficiently, you could get up to 20 bonus points on your test, not including the test corrections you'll be able to do for exam 2. So it behooves you to study in advance. So that should be your homework for this week. But that's pretty much all I have. Again, if you have any questions, please shoot me an email, and I will answer accordingly. Okay? Have a great day, and happy Founders Day.